Okay, so let us start. We are now going to be doing our webinar on how to kickstart your project, your RNA-seq project. We're going to be relying on the T-BioInfo platform, and I want to just mention here in a few words a little bit of an introduction. First of all, this project is sponsored by Pine Biotech, Tauber Bioinformatics Research Center, and the University of Haifa. We are doing a recording of this workshop, and part of the reason that we're doing this workshop is because our current users on the platform have some questions. We want to make sure that this recording is going to be available online for anybody who has any questions about it. But also we want to keep working on this format of workshops that allows our users to effectively analyze their data. Today's presenter is going to be Stepan Nersisyan. He's a master's degree student in mathematics, and his professor, Dr. Vladimir Galatenko, who's also the chief scientist at the Tauber Bioinformatics Research Center at University of Haifa. Now, before we start, I want to just mention that we have the T-BioInfo platform. The T-BioInfo platform provides a user-friendly interface for all types of omics data. And what we want to show today is just the RNA-seq data processing section and a little bit from the machine learning section. Now, what we try to do in this platform is make a user-friendly interface that provides suggestions along the way and reduces the amount of mistakes that the user can make. We also incorporated a comprehensive machine learning toolkit, and everything is organized into logical sections, as well as produced some interactive outputs. And just a side note, the platform is extendable. It is fairly easy to add new scripts and new functionality to the platform. And as we get requests for specific features, we are working to update the platform and make it more useful to the people that want to get insights from these big data sets. Now, I want to also show you a few things that we will be covering today. Okay, so unfortunately I don't see my screen. Hold on a second. Okay, there we go. Okay, so let me change my presentation. So before we start, I'm just going to give you an overview of a few things that we have been working on to make this platform more useful. So the first thing that we have is in parallel to the platform itself. In parallel to the platform itself, we produced a number of courses on transcriptomics, and a lot of the things that we'll be covering today are going to be available online for free for all the users. And you can see under courses a number of courses related to transcriptomics. So transcriptomics 1, 2, and 3 give a full overview of how to process raw data, how to do simple things like differential gene expression, and then also how to apply machine learning tools to analyze the expression tables. And we have a few other courses and a number of other projects that we are working on to make them available online. Now, we also have these workshops, and these workshops are focused on specific topics. You can see some of our previous workshops that we did this month. We are also planning a summer bioinformatics series of workshops in collaboration with a number of universities. So if you would like to join, you can email Sahil or myself. My email is ilya@pine.bio and Sahil is sahil@pine.bio, or you can use the chat box on the website right here and communicate with us directly. Now this part of the presentation is over, and let me transfer the presenter right to Stepan, who will take over from here.
Okay, hello everybody. Now I'm sharing my Okay, here's the presentation. Do you see it? Is it available for you?
Yes, we can all see it. Thank you.
Yeah, great. Today we will discuss some approaches to the analysis of RNA-seq data. Here is what we will discuss. First, we will discuss questions how to produce some expression level tables from raw fast RNA-seq data. Then we will talk about differential expression analysis, and then we will talk about unsupervised analysis and supervised analysis for processing RNA-seq data. And next slide. Let me start from definition of RNA-seq data. Here is the genome, and we have, for example, 3 genes, gene A, B, and C. And in real organism or in sample, the result of a transcription process is a transcriptome, and gene A went to the transcript A, gene C went to transcript C, and we want to estimate expression of the genes in the sample. And what's the technology? First shattering was applied. So we cut these transcripts on many small pieces. Then some adapters are ligated to these pieces. It's needed for further steps of reading. And the next step is PCR amplification. And after read, these small pieces are read by the sequenator. Yes, and so the result of this procedure is a huge list of small pieces, all just words in A, T, G, and C alphabet. And after that, we can map these small pieces to the real transcriptome. And here is the general scheme. Gene, transcript, reads, and then reads are mapped to the real transcript. And we can estimate expression level of the gene just by counting number of reads that cover the transcript. So here we go. Quantification of expression levels. One of the measures is just number of counts on the transcript. On the other hand, this approach is not so good because first transcript can be much shorter than the second. And if we will just count number of reads on it, we will get equal expression levels, but logic says that if one transcript is really shorter than another, so we need more reads to cover the longer transcript to get same expression level. So the row counts, row coverage is normalized, and 2 other measures for expression levels exist. First is called FPKM, and second is TPM. So today we will use FPKM, but TPM is also used. And all these measures can be in linear scale or in logarithmic scale. Often logarithmic scale is used because it's more useful, and we will see both cases in further steps. And let us just create a pipeline for creating expression tables from the raw RNA-seq data. Now I will focus on my browser. Hope you see it, and hope you are all logged in the platform. Please let me know in chat.
Yeah, so right now I don't see, let me see. Okay, yeah, so I see now the platform. Yeah, please let us know if you already got your platform credentials. If you are an observer and you're just watching this to learn, This platform is available. You can contact us and request access to the platform, and we can continue talking about that. But all the people that joined and registered ahead of time were supposed to get an email with their platform details.
Okay, so we see this huge platform, and we can select RNA-seq section. And we see the graph, and we can make some pipelines for analysis of our data. And first we should select some options. First we should select organism. Today we will use Homo sapiens 38 version of genome, and paired and reads. Then we upload files, and here we can directly upload file from computer. You know that sequencing files are usually very big, so it will take much time. And we prepared some links to our server, because do you see this dialog of uploading file now or not? I clicked to bulk upload and
yes,
don't know.
yes. Mm-hmm.
Great. Now I will find. Here is it. We have main files folder, and here's a file with extension.svl. All the files are already uploaded to our server, and then we click define groups, and here are our samples. There's 11 samples. Reads are pair n, so each sample is presented as 2 files. And in this experiment, we don't need some groups. It's just we will create expression level table for each of the samples. So we click no groups and start creating the pipeline. The pipeline always starts from start button. Then we need some pre-processing. As I said, first adapters are ligated to the reads. So after reading, we need to trim these adapters. And for this, we have a job, this one. And the second is cleaning some duplicates, which can appear after sequencing procedure. So these 2 buttons are just for pre-processing the data, just for trim unused things from reads. And then we need to map these row reads on the transcripts. For this, we have section mapping on transcripts, and we will use today bowtie 2 for doing that. Just clicking here, so data will start from start, then for this job, then for this, and then for bowtie 2. And then we click end, save. Then we give some name to the pipeline. Okay, expression level quantification, and save. After that, we can click run pipeline, and it will be evaluated. But our files is really big, and evaluation will take some long time. So please don't click run pipeline now. We already evaluated it and have some output results. Let me focus on it. Yes, now you see my folders, and here's a folder with results. Let me open it with Excel. Now I need to focus on Excel. Okay, now you see the table. This table is a result of expression level quantification. Columns are samples. Here you see 11 sample names, and rows are genes. You see a big number of genes, and for each gene and for each sample we have a expression level in oh, I forgot to, I'm sorry, in this pipeline I forgot to, after mapping we should select, 1 second, I will do it one more time. We should select job for estimating expression levels. 1 second, I will focus here. Just 1 second. So do what we have done. First this, then this, then mapping. Yes, and then we want to make expression level table. And for this, we have a job called RNA-seq table. So we should select it. And here are 3 measures, 3 units of expression. And we should select FPKM today. And then we could select end. Sorry, forgot it last time. Correct it. Yeah. And this is pipeline for obtaining expression levels of the genes. Let me go back to the resulting table. Here is it. And for each gene and for each sample, we have a number, and in FPKM units showing level of expression of the gene. You can see there are some 0 lines. It means that gene is not expressed in all samples, and you can filter out these rows by simple filter in Excel. And we have a already filtered file. Let me open it. Here is it. It takes some less space. I can show it to you. Yeah, here is it, and you can see there is no 0 rows, and it is a list of genes which are really expressed in our samples. Yeah, and what can we do with this data? Because you can see there's many rows. Actually, it's 20,000 rows, many numbers. And first problem with which we can solve is the differential expression analysis. What is it? We have some samples, and they are divided in different groups. And we want to understand which genes are expressed in one group and not expressed or less expressed in the other group. So differential expression. For it, we will look at the same data sets. Let me go back and create Warmo pipeline. We should select human paired and upload same files. And now we can define the groups. We have now 11 samples. Actually, it's some cell lines with and 1/2 of them are with cancer. So we can create here one more group by pressing on plus and drag last 6 samples to the second group and first 5 samples to the first group. So now the system knows that these samples are from first group and these from the second. Then we press continue, submit, and create the pipeline. Pipeline is very similar to previous one. So we start from the same pre-processing. Then we select mapping with bowtie2. Then we are creating expression tables. And then we want some differential expression analysis. For it, we have some tools. For example, we can select edge R or dissect to. Let's select edge R and go to the end. Name the pipeline. Yeah. So what we expect from this pipeline? Let's go back to the presentation. Okay. So what we expect from it? First, we expect a difference between mean expression levels. This value is called fold change. And you really need to pay attention to the scale. Is it linear or logarithmic? Because 1.1 in linear scale is nothing, but 1.1 in logarithmic scale means the difference in 2 times. Please note that big fold change is not enough for saying that we have really different expression levels. Let's look at these 2 pictures at the right of the slide. We see 2 genes with their expression levels in the samples. So first group is a blue one and second group is a red one. And in both pictures, we see big fold change. In the bottom picture, it's obvious. The means are distinct. In the top picture, it may be not so obvious, but you see that the top points, which are blue, we have many blue points on the top, so mean of the blue group is located higher than the mean of red group. So we have really not zero fold change, but it's obvious that on the top picture there is actually no difference in expression levels between samples. It's some artifact that these blue ones are such near to each other, and on the bottom one we see really significant expression difference in expression levels. So for formalizing these things, there exist several tools which aimed to the analysis of variance of these sets, and they all result in one number, p-value. So p-value is a probability of observing such or higher difference if we assume that there is no difference in samples. So small p-value indicates that we have statistically significant difference between means of the groups. But I showed that we have a really big number of genes, 20,000, yes. And when you do multiple testing, you should adjust p-values. Let me introduce some example. If you will toss a coin 10 times and will observe 10. One second. I'm sorry. If you will toss a coin and observe 10 highs, you will think that this coin is not normal. But on the other hand, if you will give a coin to all the people from Earth, several billions of people, obviously there will be some big number of people who had rows of 10 same results. So when you do multiple testing, you need to adjust your p-values. And adjusted p-values sometimes are called fault discovery rates, FDR. there are another approaches, but we will look only at FDR today. And the third thing that we should note that sometimes we can see a gene with statistical difference in statistical significant difference in mean levels, but these genes are low expressed. So we are comparing 0 with 0.001, and you should also note that it's not meaningful to look at these positions. The pipeline I've created is will also run very, very long, so we have already prepared result for it. It is in diff expression folder. We can open it with Microsoft Excel and see how it, oh no, it's not, yes, here is it, and see how it looks. So our rows are gene names, genes, and we have 4 numbers. First is logarithmic FC, fold change. So I have already said that it's important to have logarithmic or not logarithmic. So logarithmic fold change equal to 10 means difference in 2 power 10 to 1000 times. Second column is CPM. CPM is a characteristic which indicates expression level, mean expression level of the gene. So as I said, if this value is really low, it is meaningful to remove corresponding gene and don't look at it. This column is p-value, and this column is adjusted p-value, false discovery rate. So now we can filter this table just by false discovery rate less or equal than 0.01. I understand my Excel is in Russian, but I command this. So yes, now I have filtered that false discovery rate is less than 0.01. And second filter is that logarithmic fold change is between 2 intervals. So in one hand, it will it need to be greater than 1. It means difference in 2 times or more, or less than -1. It is difference in 2 times in the other side. So greater in 2 times or lower in 2 times. And here we see how many significant positions we have. About 700 of genes are with really statistically significant difference in mean level of expression. Yes, so it's AGR, and there is another tool, DSEQ2. One second. Yes, we have result of DSEQ2, and you should note that the results of 2 different tools can be essentially different. So they use different statistical frameworks, and results can be different. For example, DSEQ2 usually gives more significant positions than HR. Yes, and now we as I understand, one second. Okay, now we created the pipeline, and I think that's all about differential expression level. Next, we will. Okay,
so
I see question.
Stepan, yeah, okay, I just wanted to point it out.
No, so sorry. How to filter the genes with low expression? What cutoff do you use? This question is difficult, and sometimes I think that you should look at corresponding positions in separate. So I don't think that there is a general approach to cutting off. So you can draw, for example, histogram and see and take a number of points with really low expression in comparison with other genes. So I don't think that I can say that there is a unique threshold for all problems. And
but in general, when you mentioned filtering the expression table, so you filtered out the zeros, right? So those genes that are zero across all of the samples.
Yeah. So I have removed them. Yeah.
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 And then when you say the low expression, I mean, you didn't filter anything in your table, but would you do something with your table before moving on to the next step?
I think that 0 rows are cut by HR2, so I don't think that we need to do it in the manual way. So in table which I showed, 1 second. In this table, there are no positions with really 0 mean coverage, expression level, but you can see that there are really positions with negative expression level in logarithmic scale, and I think that looking at other numbers like 4, 6, we can say that, for example, this gene is the result of low expression, and we could remove it and don't look at it. So if we see that other numbers are relatively big, we can estimate cutoff for current problem. So I don't think that there exists one unique cutoff for all tasks in our world. Okay. So if there are any more questions, please feel free to ask them in the chat. And there is no, we can go further. And we have some question from the previous workshop. The question is how can we work with not human organisms? And you see that in the T-BioInfo platform, we have a lot of other organisms except Homo sapiens. And if you have an organism which is not listed here, you can choose your genome and upload it too. So this platform is aimed on an arbitrary genome, arbitrary organism, and you can work not only with human. Okay, if there's no questions, we will continue. Okay, and now will we talk about how to generate SVL links today? SVL links is a links to the files on our server. So for obtaining this link, you should upload the file to the server and ask someone from the administrators to send this link to you. So this is just our current preparation. We've already uploaded it to the server, and I think I've asked your question. So today we will not upload any big files and get SVL links to them. Okay. So SVL link is just a path to the file on the server, not something special. Okay, and let us wait some seconds if there are more questions. Okay, let's continue and let's speak about unsupervised analysis. In previous example, we have 11 samples with which are already separated into 2 groups. But sometimes we have some samples and we do not know if there exists some partitioning groups which can be really meaningful. And this is a topic of unsupervised analysis. So we have some number of samples and we want to understand, can we divide it into some groups to see some meaningful partition. And I will demonstrate you 2 approaches. And the first approach is a principal component analysis. For accessing it, we should select unsupervised analysis section here. And what is principal component analysis? Imagine that we have only 2 genes in each sample, only 2. Just funny situation. But if there are only 2 genes, then we can draw each sample on the plane, just 2 coordinates, expression of the first gene and the second gene. And we have some kind of, I don't know, 10 or 20 samples. We can draw them on the plane and see these stands are above and these stands are at the bottom of the plane. And we see natural division to the groups. But as you've seen, we have 20,000 genes and it will be difficult to draw them in plane, but we will take some projection. Let me share my webcam and demonstrate what is principal component analysis. Now you see me, and as you know, I am a 3-dimensional object, but actually you see my 2-dimensional projection on the plane of your screen. And let's take a pencil. Now you see the projection of 3-dimensional pencil. If I will orient it like this, you see just some little circle, so you don't see nothing, and you can say that this is a pencil. On the other hand, if I will turn it this way, you see that this is actually a pencil, and you see that it's a circle and it's some kind of segment. So the idea is to maximize the variance of our set at the projection. And what does the principal component analysis do, but for the high dimensional case? So let us upload some files. We will upload expression table. There is a main files folder, and we have here cell lines expression data. Now we upload it by simple upload, not SVL links. Wait for it being uploaded. Then press continue, start. Okay, PCA draw. And here's just some default arguments, and then go to the end. Save and PCA. And after that, we can run the pipeline. it's not so long as the other pipelines like mapping or obtaining expression table, but let us go to the results, not to waste some time. And here is PCA folder, and we have a PDF, the picture which the job gives. Now I will show you to it. Show it to you. Okay, here's the projection of the data to the plane, and you see that in the left we have one group, and the right we see the second group. So PCA is just a method to see by your eyes. So it's not automatical procedure. You just draw and look at it. And in this case, it's obviously separation into at least two groups. We could look at first and third principal components. Here we see the partition two, and second and third. I don't know if you can see something in this picture, but okay. And one more case, when you have some partition to the groups and you want to understand if they are really distinct. So, for example, we have input file, expression data marked. We can open it with Excel. Here's a table with huge number of samples. We have about 50 samples, and each sample is marked as some group. So it's some cancer cell lines, and you can see some groups. And we want to visualize it by principal component analysis too, but we want to see the plane with points colored by the original groups. You see our pipeline already finished, and here how it looks like. Here are results. You can download this PDF, open it. Yeah. Okay. Now we will run another one pipeline. Oh, 1 second. We go here, start. Okay. I forgot to upload some files. Files marked. Okay, we just repeat the same procedure. So start, PCA, draw, and end. And not to waste time, I can show the result. Don't think that it's important now to see the process one more time. And here is the result. Here we see the 4 groups, and we see that one of the groups, the luminal, is really separated on the plane of first 2 principal components, and the 3 other are just mixed up. At the plane of first and third principal components, we see that actually basal group is only distinct with other 2, and on the last one we see nothing, and it's normal. Second and third component does not contain a lot of information, such the first principal component. And that's about principal component analysis. And for your job, you may need to modify this plot or add something new. And you can see that in output files of this pipeline, we have something called pca rpack.zip. we can extract it.
Sorry, Stepan, I see your PowerPoint or your PCA platform.
I'm sorry. Yes, here I focused now to the folder. Sorry. We have an archive, PCA-R pack, at the output of the pipeline. We can extract it. And here we have our script, which performs all the job. Let me open it with. Oh, open Notepad is not good. 1 second. Okay, I will open it with Microsoft Word. It is opening now. Okay, and here's the code. And for example, you may want to add a title to the picture, and you can find here the line which performs plotting. It's called ggplot function. And you can add there something like labs title equal to, I don't know, some hello world. And if you will run this code, you can see that in this folder we have, oh my God, 1 second. I will close this. We have one more file. Let me open it with Notepad. And it contains information about how to run this in with R. So here are arguments, just input file, and 4 arguments which are need to be passed to the PCA. Okay, so you can modify the script and obtain some other results. Where's my presentation? Ah, here is it. That's all about unsupervised analysis. And the next problem is supervised analysis. Here's some slides about hierarchical clustering. It's an alternative to the PCA, which results in the following picture. Here is some instruction how to run it. Just start, hclust, and here's some options. And the result of this procedure is dendrogram. It's a tree. Height is the rate of dissimilarity of the samples. So the more down you go, the more samples are similar. And from this dendrogram, we see that we can speak about 4 clusters. For example, this is the first cluster. First group and cluster are synonymous. This is the first, second, third, and the 4th one. Next are not meaningful because they are really similar to each other, and no need to separate them into different clusters, different groups. And now about supervised analysis. What is supervised analysis? Let me show you input files, and it will be much easier to understand it looking at these files. Suppose you have some samples which are divided into groups. Let's look at the same table. We have some samples and some 6 groups, 6 classes. And then suppose we have one more sample and we do not know to which group it corresponds. For example, let's open test file. And here we see 2 samples with their expression levels, and we do not know to which group each sample corresponds. And we want to train a classifier on previous dataset and evaluate it to make prediction on these 2 samples. For that purposes, we have let me focus back. we have a supervised analysis section in the platform. The workflow is similar to previous ones. We just upload 2 files, train and test. Wait until they will be uploaded. Okay, then press continue and start the workflow. Here you see several methods. Today we will use SV stepwise LDA, linear discriminant analysis, because our tables contain a lot of features. 20,000 genes is much bigger than number of samples. So naive applying of some other classifiers like random forest or SVM will cause in not meaningful results, just some meaningless classification. But stepwise LDA performs feature selection inside of the algorithm, so it really can work with our data. And here we need to modify one of the thresholds of the algorithm, set it to 0.005. Oh, just add 1 0 in the after decimal point. Click save and go to the end. We can give it its name, and after that we can run this pipeline. Stepwise LDA will take something about 5 minutes, so I think I can show you the results now, not to wait until it will work. Yeah, and let's look what predictions it made. Predictions are in SVLDA prediction test file. We can open it via, for example, Excel. Yeah, and here we see sample name and predicted class. I have removed these 2 samples from the previous big table, and actually I know which classes are correct, and I can say to you that these predicted classes are correct. So classifier resulted in correct answer in both cases. So this sample is really normal-like, and this sample is really basal. Yeah, so this classifier really works. Now I will go back. And the last thing that I want to discuss, let's open this file with coefficients. Yes, these are some genes which stepwise LDA extracted as the most meaningful genes to making predictions. So here we see some ensemble IDs of the genes, and if you are a normal human, you can't understand which this string means. Yes, you need some description about this gene. And first approach is just copy this name and paste in Google and see what is it. Or the second approach, the normal approach, is to go to the platform, go to the utilities section, and select here annotation button. We can upload the file. Where is it? Where is it? Where is it? We can upload the file, press continue, start, annotation, and the end. Gene, gene, okay, we can run it, and the result of annotation will be a table. Let me show it to you. Yeah, here is it. We can remove now these columns. We can remove them. Yeah, and we have a table with annotation, just all information which you can find from the Ensemble website, just GenSample, some short description, and other. So all information about gene by its Ensemble ID. Okay, and have it finished. You see that SV LDA is processing now, so it's good that I showed the results already. Okay, so now some summary. What we have done today. First, we have analyzed some approaches to obtaining expression level tables. Then we spoke about differential expression analysis, about DSEQ2 and HR, and then we talked about supervised and unsupervised analysis. If we do not have an additional information about groups, we can apply unsupervised approach. For example, PCA visualization and looking just to the picture, or hierarchical clustering, or some other kind of clustering. And if the samples are divided into groups, we can perform supervised analysis to understand to which class, to which group new sample belongs. Yeah, I think that's all. And you can ask questions.
Yeah, so please ask questions. We want this workshop to be also an opportunity for you to start your own projects. So I know that there are some technical questions that come up as you try to upload files, questions around what formats you are able to upload and what parameters to set in some preliminary steps. So this is the time to ask those. Also, there might be questions about some non-standard ways to process data. So this was an overview of a standard RNA-seq. You can do a lot more with next generation sequencing data, but we can cover that in separate workshops or in one-on-one conversations. We also have these different sections like supervised and unsupervised machine learning, and there are a number of other methods there, and you're welcome to try those just to capture the difference. The difference is that in one you process raw FASTQ or FASTA files. In the second one you work with tables, and I think that was demonstrated in the workshop. Also, as a conclusion, you get some visualization in some of the algorithms. Not all of them produce visualization, but typically you can open a table in Excel and do some kind of simple chart in there. And so we saw a little bit of that also today. Hopefully it's made it clear how to get started and how to upload your files. If you need help uploading large files of a lot of samples, if you're already using the platform, to keep reusing the files. So let's say you started one RNA-seq pipeline and then you want to try another differential expression pipeline. It's very helpful to have those files as SVL links, because then you can reuse them and don't have to upload them many times. Just contact us and let us know about it. Right now we have a number of users on the platform, and we are monitoring how much space and how much resources are being allocated towards each project. And so we help minimize the mistakes and different technical issues that come up by helping to make sure that everything is uploaded and stored in the right place. Okay, and then okay, yeah, so Cheng is asking about fastq.gz. Yes, you can upload those now. They will work fine. Okay, so the fastq.gz files get unzipped inside the begin folder now. And also, it would be great, again, just as a reminder, if you have more than several samples and you want to have access to those samples in many different pipelines, it's good to upload them, and we will provide you with the links to those files, SVL links. And also, if you want to take a look at some of the details of what we use today, some of the links that we used, all of these links, so again, these are real files with SVL or tables. All of those are located on the workshop page. I will paste the link here once again so that everybody has the link. And on this link, okay, sorry, let me make it to everyone. So on this link, if you scroll down, you can see files for download, and there's an orange button, download file. So you can just download this. Here, let me quickly share my screen. Okay, so as you can see my screen, this is the link I gave you. You can go down here, you can download the file, and you can also download this presentation. So when you go to click on download file, you will see the workshop file zip, and within the zip you have the presentation, you have the input files, and these are the SVL files, and then you have all of the output files. All of this is just to help you get started. If you want to play around with the platform and you have the credentials to access the platforms, it's very simple. You use upload, and then you can use some of these files to just get started. Now, you also can download files from something like Geo, or if you have your own files. So if you have any questions about that, just let us know. All right, well, I hope that this was helpful. We will continue and expand on these workshops in the near future. We are planning a more comprehensive hands-on series of workshops that we will also schedule in person. And I want to just conclude with a couple of dates. So in May, around the middle of May, we are going to have hands-on workshops in a number of places in the US. We will also offer those online. And then we also will have a summer workshop, a summer series of workshops. And in the summer, if you can let us know that you're interested, we are still planning the locations. We're thinking to start this around June. So please reach out to us. That's about it. If you don't have any other questions, we are going to end here. So again, thank you to Stepan for sharing the slides and going through the steps. And thank you for everybody else that attended. And please stay in touch. Thank you.
Thank you.
Thank you.